going to discuss about life cycle of HIV. Okay, so first of all, as we know that CD4, we can say T helper cells contain the receptors which are CD4 receptors. So what is the work of HIV? HIV basically infect or destroy that CD4 cells. Okay, HIV basically what it infect or destroy CD4 cells. Okay, so when we can say the CD4s are infected, so there will be no B cell or T cells activated. So now let's say the HIV infection is basically carried out in following steps. So if I can talk about the first step, so that is absorption or attachment. First step is adsorption, yeah, attachment. First step is what? First step is adsorption or attachment. So if I can talk about this step, so here we can say that viral basically particle attached on the surface of lymphocyte cells. Okay, so as in the previous video we have discussed that GP120 attached. So it attached on what? It attached on surface of lymphocyte cells. Okay, it attached on the surface of lymphocytes. So when this is attached, what will happen? This will allow the CD4 receptors of lymphocyte along with co-receptors. Now, as we have discussed now. Now let me tell you when this is attached. So we can say CD4 receptors are basically attached on the lymphocytes, and lymphocytes basically contain the co-receptor that are CCR5 and another is CXCR4. These are basically core receptors in which these are core receptors present in lymphocytes. Okay, so if I can talk about particularly, so this is basically CCR5 is commonly present on macrophages, dendrite cells, and these all macrophages, which are type of lymphocytes, dendrite cell. Okay, but if you talk about CCR4, so this is not commonly present, and this is basically the receptor which is present on T helper cells. T helper cells. Okay, so now let's move further on. So yeah, when this is basically attached to viral envelope glycoprotein, which is GP120, it will bind to the CD4 receptors of lymphocytes. So if I can here make a cell, and here we have CD4 receptors. Okay, this is CD4 receptor. Okay, so now here we have what? One glycoprotein GP120 which come and it will attach to this, and GP120 have the receptors will attach on this CD4 receptors. Now here we have what? This is, if I can suppose this is a macrophage, so it have basically co-receptor which is what? CCR5, which is co-receptor CCR5. Okay, so yeah, now this is how this is attached. Now remember this, the receptors of this lymphocytes, and we can say GP120 will attach, or it will attach by the mutual affinity. Matlab it will not be removed, attached by mutual affinity. It will attach by mutual affinity. Okay, so now viral particle is basically completely attached with the receptor of, we can say, co-receptor. Now here we can say, if I do talk about the co-receptor, so this was CCR5. So this viral particle will completely attach to the CD4 and CCR co-receptor. After the contraction of, after the, we can say, attachment or contract of this, we can say, this receptor, we can say GP120 receptor and this co-receptor CCR5. So now the fusion will begin. Now remember that CCR5 co-receptor is present in GP120 transmembrane. So after when, now if I can write here, so receptor of, we can say, viral protein, receptor of viral protein, and we can say co-receptor attached. Okay, so when this co-receptor is attached, so the next will be your fusion will be, okay, so fusion will start. So now here down, let's see the second step, which is fusion. Okay, so this fusion is basically after attachment the viral envelope, then we can say connected with the host cell membrane. So now in the fusion, what will happen? The viral attachment, you can say viral envelope is connected with the host cell membrane. It's connected to host cell membrane. Connected to host cell membrane. Okay, so now when this is connected, so this process is known as mediated and by other viral glycoprotein GP140. This fusion will occur, we can say, cell membrane, the GP120, oh sorry, GP140. Here we have GP41. So what will be the work of GP41? So as I told you, GP41 basically what, transmembrane protein. So this is the transmembrane proteins, so we can say of the viral envelope, and which facilitate what, which facilitate the fusion of viral envelope with the host cell membrane. So this means कि viral envelope और host cell membrane की क्या हो गई, fusion हो गई. Fusion of viral envelope with host cell membrane. Here if I can draw a picture for you guys, so let's see. mm, yes, so here I'm going to draw. Yep, for this suppose we have this one HIV virus. So HIV virus basically contain spikes. Here we have double membrane, lipid barrier, and here we have GP41. Here we have GP120. Okay, and now this is what we have, another cell, macrophage T helper cell or dendrite cell, and here we have receptors are present, which are CD4 receptors. Okay, CD4 receptors. These are CD4, and this is what GP120. So when these both will attach together, so what will here, the fusion process will occur like this. If this is what, this is the cell, so this will be what, like this. Here we have spike. Okay, and here we have what, here we have, this is the fusion occur, and now your genome is present here. Baki remaining part will be outside of this. Okay, so this means genome is now inside. So until that it is clear to you guys. Now we are going with Basically, the enzyme here which will be used, which is reverse transcription, will happen here.